A'udhu billahi minash shaitanir rajim. Bismillahir rahmanir rahim. Dear students of 12th class, Boys Higher Secondary School, Nagari, of subject Botany. Assalamu alaikum wa rahmatullahi wa barakatuhu. Dear students, you know that we are studying the development of endosperm, and in the previous lecture yesterday we have discussed the first type of endosperm, that is the nuclear endosperm. And you know that there are three types of endosperm formation. One is the nuclear type of endosperm, second is the cellular endosperm, while as third is the halobial endosperm. The first one, that is a nuclear type of endosperm, we have discussed in our previous lecture yesterday. Now today we will discuss two more types of endosperm. One is the cellular endosperm and second is the halobial endosperm. Now regarding the cellular endosperm, this type of endosperm formation occurs in plants like datura and petunia. These are the two types of plants in which this type of endosperm formation takes place. The names of the plants are datura, जिसमें इसको हम कश्मीरी में दतुर बोलते हैं, and second is the petunia. So this type of endosperm formation occurs in the plants like datura and petunia, etc. The endosperm nucleus shows continuous divisions. The continuous divisions occurs in the primary endosperm nucleus, and those divisions are followed longitudinally or transversally. That means two types of divisions occurs, either longitudinal division occurs and or either transverse division occurs. So the primary endosperm nucleus shows continuous divisions either longitudinally or transversally with the formation of cell walls, resulting in the formation of cellular type of endosperm. So dear students, here you should remember that this type of endosperm formation occurs in plants like datura and petunia, and the primary endosperm nucleus shows the continuous divisions longitudinally or transversally with the formation of cell walls and resulting in the formation of cellular type of endosperm. Dear students, here you can show the diagrammatic representation as this is the first diagram of development, this stage is in the development of cellular type of endosperm. This is the first diagram here. This is the embryo and embryo sac, and here it is a second, this diagram, second figure, and you see here the wall formation. And here these are the embryos here and the embryo sac, and ultimately this is the cellular type of endosperm, this figure D, and this figure A, figure B, figure C, and figure D are the stages in the development of cellular endosperm. Now, the third type of endosperm is the halobial endosperm. Halobial endosperm. This type of endosperm is an intermediate type of endosperm. This halobial endosperm is an intermediate type of endosperm which is formed in monocots. Dear students, you know the dicots and the monocots. Dicots have the two cotyledons, as well as the monocots have the two. This have a single cotyledon. So this halobial endosperm is an intermediate type of endosperm which is formed in the monocots. Here what happens, the primary endosperm nucleus moves to the chalazal end of the embryo sac. Dear students, you know There are two ends of the embryo sac. One is the micropylar end and another is the this chalazal end. So here the primary endosperm nucleus first moves to the chalazal end of the embryo sac. After that, the first nuclear division occurs and a transverse wall is formed, resulting in the formation of a small chalazal cell and a large micropyle cell. So, dear students, what happens here? The primary endosperm nucleus first moves to the chalazal end of the embryo sac, and after the first nuclear division, transverse wall is formed, resulting in the formation of two cells. One is a small chalazal cell and a large micropyral cell. The chalazal cell functions as hostorium. The chalazal cell functions as hostorium, whereas the nucleus of micropyral cell divides by repeated pre-nuclear divisions. So here you have to remember that the chalazal cell functions as hostorium, while as the nucleus of micropyral cell by repeated pre-nuclear divisions, the nucleus of micropyral cell divides by repeated pre-nuclear divisions. The nuclei now get scattered around the periphery of embryo sac. वो छ गसान यम embryo sac side manz yum nuclei roza. Formation of cell wall now occurs. Now the formation of cell wall occurs and a cellular tissue is formed which fills whole of the embryo sac, leading to the formation of halobial type of endosperm. So this was all about these two types of endosperm, cellular endosperm and halobial endosperm. These are the diagrams of this halobial type of endosperm here. First you see when the division occurs here at the top end, at the macropylar end of the embryo sac, there is a large micropylar cell, and here it is the small chalazal cell. So this is the nuclei at the periphery of the embryo sac. यहाँ पर आप देख सकते हैं side में at the periphery of embryo sac. These are the nuclei which get scattered, and these are the free nuclei here, and this is the micropyle cell, while this is a chalazal cell. And these four diagrams on this first, second, third, and fourth, A, B, C, and D. These are the stages in the development of halobial type of endosperm. So this was all about today's lecture. This development of endosperm, cellular endosperm, and halobial endosperm. And in this way, the development of endosperm we have completed today, and you now get aware that there are three types of endosperms. One is the nuclear endosperm, which we had discussed in our previous lecture, and today we have discussed these two types of endosperm. One is the cellular type of endosperm, and the third one is the halobial endosperm. Now we have come to an end to this topic, today's topic. Stay home, stay safe. Thank you very much.